video I'm going to be talking about microscopes, and this is basically the theory section of all the three parts. So the usage of microscopes is the first thing that we're going to learn about today. Microscopes, they use something called lens. Now what are lens? They are basically pieces of glass which help to actually magnify certain images, and it also allows you to see certain images in focus, which is kind of what resolution is all about. So they allow objects to look bigger, and they also allow these objects to be focused and you can see them in detail. So if they ask you what the definition of resolution is, then resolution is basically the distance between 2 points which can still be seen as 2 points. Okay, so sometimes when 2 points are so close together, they can sometimes be seen as 1 point, which is like at the bottom. This is because the 2 points are so close together that the lens in my camera can't really detect them. Whereas these 2 points can still be seen as 2 points, so this distance is detectable by the camera. So the camera has the resolution to detect these 2 points as 2 points. So that's basically what resolution is about, and this is the definition that you need to learn if they ask you the question in the exam. Now, so that's basically the use of certain microscopes. They increase the magnification, they increase how big an object looks, and they also increase the focus of a certain object. Now, part 2 is about microscopes. Microscopes can be light microscopes or electron microscopes. Light microscopes, a bit of context, they were made in the 1590s. What do? They pass light through a specimen. So say, for example, if you have a certain specimen on your stage, then the light microscope will pass a light ray through your specimen. What does it allow you to see? Things such as nuclei and chloroplasts. So, oh my gosh, the tea has been wiped out. But anyway, so what are these things? They are rather larger internal structures of certain cells, so they're larger cellular components. We also have something called electron microscopes. So electron microscopes were created in the 1930s. They use electrons, so they use electric beams which pass through the specimen. The magnification and the resolution is much, much higher of an electron microscope than there is of a light microscope. So therefore you can see things in much greater detail. You can see many more things in much greater detail. So for example, the internal structure of mitochondria. First of all, mitochondria themselves are very small cellular components. Now if you can even see the internal structures of them, that shows how more powerful the lens of electron microscopes are than of light microscopes. The mechanism behind electron microscopes is they allow you to see how certain processes work of the cells and the role of the certain structures. So they allow scientific knowledge to expand, and it's what discovery nowadays is dependent on. Now, just one more thing about resolution, because it does seem a tiny bit harder. Here, if you have, resolution is always fixed. So of a certain lens, the resolution is fixed. It's just the 2 points, the distance between the 2 points, which can be different. For example, if the resolution is 2 centimeters, that basically means that if it's either more than 2 centimeters, if the distance between the 2 points is more than 2 centimeters or 2 centimeters, it can still be seen as 2 points, like shown here. But if it's anything less than 2 centimeters, if the distance between the 2 points is, for example, 1.9 centimeters, what do you see? You just see one big blob and you don't see 2. That's because it is much lower than the minimum resolution. So that's basically what the whole theory of microscopes is about. Hope you liked this video. Please don't forget to like, subscribe and share. Bye.